I'm a bit nervous. There's a big hard act to follow. I realise I'm all that's standing between you and morning tea, so I'll only take up about 10 minutes of your time. Thank you. As Peter said, I'm working at Trobe University. I'm towards the end of my PhD, and this project originally started off as a project to investigate improvements to salinity tolerance. But what I'm going to talk about today is this really interesting and exciting and unexpected finding that we have, which is related to, beg your pardon, there we go, to seed protein accumulation. And that's what I'm going to talk about today. So just to give you a bit of background to where this project started, as we know, one of the major issues affecting Australian agriculture is the increasing salinity of otherwise highly productive land. Salinity refers to a high concentration of salts in the landscape, to the point that they have a negative impact on soil and water quality. And the main culprit is this guy, this sodium chloride. The costs to agriculture are obviously significant, and that figure doesn't just reflect crop losses, but it also includes things like social and environmental impacts, changes to water quality and biodiversity, things like that. It does occur naturally over time, but it's exacerbated by specific human activities. And as we've come to accept over the last decade, there is no quick fix, unfortunately. What's actually happening in the plant, salt causes a reduction in crop yield and quality when it accumulates to toxic levels in the shoots of the plant. So sodium in the soil, as I said, the main culprit is this guy, this sodium ion. So he accumulates in the soil and then enters the roots passively, gets carried up the root to the shoot where it's deposited, and then it accumulates in the cells to these toxic levels. Now, one way that the plant has to cope with this is it uses these red guys here, these sodium pumps. What they do is they pump sodium out of the cytosol where it's toxic and into these internal compartments where it's kind of safely stored away and effectively detoxified. And what my project really started off looking at was, can we use this mechanism to improve salt tolerance in our crops? The pumps themselves, they're called sodium-proton pumps, or NHX proteins for short. And they do kind of exactly what they sound like. They pump sodium and hydrogen in and out of the cell and in and out of different compartments in the cell. So our research is focused on just two of these proteins. They're arbitrarily called number 5 and number 6, so AtNHX5 and AtNHX6. And the reason primarily that we were interested in these is that very closely related protein, number 1, has a very well described role in salt tolerance. And then recent work in our lab suggests that number 5, AtNHX5 and AtNHX6, are also involved in the plant stress response. And this means that they're likely to be potentially really useful in terms of genetically modifying crops to improve salt tolerance. However, if we do want to safely and effectively use them in generating transgenic crops, it's really critical to understand the developmental role that they have in the plant. It's no good to have a salt-resistant crop that can't stand up because it doesn't have a developed root system. And so to that end, my project primarily has been looking at what else can they do? What else do they do in the plant? As I mentioned at the start of my talk, I'm interested in seeds, reproduction in plants and seed health. And so I was interested in investigating the seed protein profiles and how these genes were involved. So I'm going to take just a small leap of faith and tell you a little bit about seed storage proteins. So once again, they're named pretty aptly. They are kind of exactly what they sound like. Cells within the seeds accumulate and store these large, huge amounts of protein during development, and they're called seed storage proteins. They're important for the seedling during the early stages of germination. So up here, before he's able to put roots down into the soil so that he can feed from the soil, he needs to have a feed source. Since they make up the bulk of protein within the seed, they're also obviously very important for human and animal nutrition, and that's why we're kind of interested in them. Most importantly, and this is kind of the critical point for me, I guess, is that their correct production and storage during this phase of development is really critical for seed health and longevity. And what's interesting is we can look at them. So what I've got here is I've taken some regular, normal, healthy seeds. I've taken the proteins out of them, and then in this picture here, the proteins separate according to size. So we have some larger ones up the top here, these faint bands up here, and then obviously this big chunk of slightly smaller proteins. And in normal, healthy cells, what's actually happening is the cell makes these big precursor proteins, so it makes them big, and then it chops them up, and then it stores them. So the mature forms are these chopped up ones down here, and they accumulate to these really high levels. So at any kind of point in time, you have a few of these like transitional larger proteins, but you have this big chunk of the smaller mature proteins. Now, if we want to have a look at what happens, how the NHX proteins are involved, the easiest way to do that is to turn them off and see what happens. So what we find is that if we turn off, what I've done here, these are seeds with the NHX5 gene turned off, and these ones have the NHX6 gene turned off. And really, you don't see a huge difference. They look really similar to the ordinary normal seeds. But if you turn them both off at the same time, so this is just two different sets of seed which have both the NHX5 and NHX6 turned off at the same time, we see this massive increase in the amount of precursors that are present in the seeds at the time. Now, I think that's really interesting, because obviously what's happening in these cells is that they're still making these proteins, but they're not processing them correctly to the larger forms. So given that now we know that there's something going on in the cells of the seed, I thought I'd have a look at them. So if you have a look at them under the microscope, they're really pretty. So these are just normal embryo cells, and they have in them these big bubbles. So this is a single cell. And inside each cell, there's these huge bubbles that are just chock full of protein. So in normal healthy cells, they're big and they fill up most of the cell. Once again, if you turn both of our genes of interest off, what happens is instead of having nice big full bubbles, maybe five or six of them, instead you have like up to 30 of these tiny, misformed bubbles. They don't seem to be forming correctly. And if you do a quick count of them and measure their size, what you find is that in the normal cells, the area of the bubbles is much bigger, but there's much fewer of them. But when you actually do the mathematics and work out kind of what the total area is, you find out that they're almost the same. And in fact, it appears there might be more total volume in these guys than there is in these guys. And I guess that's really interesting in terms of kind of the total protein content of these seeds. And that really brings me to kind of the next question that I'm going to try and answer in my project, and that is there more protein than normal in these seeds, or is it just that there's a change in the ratio of protein? And possibly even more importantly, is that protein of high quality and stability? Along the same lines, and maybe this is more pertinent to the question of generating transgenic crops, what I've done in my research so far is turn those genes off. But what happens if you increase the amount that you have in there? So instead of stopping the activity of these genes, what happens if you increase it? And these are all questions that I'm going to try and answer over the next 6 months of my PhD. So I guess my main points are that NHX5 and 6 are likely to play a significant role in crop salt stress response. And if this is the case, then that would enable them to be used to generate transgenic crop species with an increased tolerance to saline environments. Really exciting. However, to successfully and safely use them in transgenic crops, we need to understand what the broader impacts are on plant growth and development. Really critical. Unexpectedly, in the course of my PhD, I found that NHX5 and 6 are important in the normal accumulation of seed storage proteins. And thus, increasing expression of NHX5 and 6 might have an effect on seed protein content or quality in addition to improving salt tolerance, and I think that's really exciting. Great big thank you to the GRDC, not just for their funding, but also for inviting me to speak today. My two supervisors, Dr. Tony Gendel and Dr. Kim Plummer from the Trobe University, and to my colleagues Brett and Partha, who've done a lot of the foundation work for this project. Brett and I actually published a paper last year describing some of this work, so it's very exciting. And of course, thank you all for listening.